Muga silkworm, Antheraea assamensis. The Muga silkworm, Antheraea assamensis, belongs to the Saturniidae family of the Lepidoptera order. Both Antheraea assamensis Helfer 1837 and Antheraea assama, Westwood, 1848, are synonymous, with the name Antheraea assamensis being upheld based on the priority rule of zoological nomenclature. This species is predominantly found in eastern India and its surrounding regions. The cultural practice of rearing Muga silkworms is primarily observed among the elderly populace of the Brahmaputra Valley in Assam and neighboring areas. The term Muga originates from the Assamese language, signifying brown or amber. Optimal conditions for the growth of Muga silkworms are typically observed at temperatures ranging from 24 to 30 degrees Celsius and relative humidity levels of 75 to 85%. Host plant: the Muga silkworm is polyphagous, primarily consuming the leaves of some, Persea bombacina, and Solu, Litsi polyantha. Other notable secondary host plants include Mejankari, Litsi citrata, and Digloti, Litsi salicifolia, all belonging to the Lauraceae family. Additionally, Muga silkworms are known to adapt to over 24 different food plants for survival in various ecological habitats, many of which are classified as tertiary host plants. Morphological features: moths, ochreous brown to dark chestnut brown in color, with a dark inner part and yellow-brown outer part in the eye spot. Male moths have a wingspan of approximately 13 centimeters, with sharply curved forewing tips and rounded hind wings. Female moths exhibit a wingspan of about 15 centimeters, with a gray submarginal line on both wings. Wild strains in Arunachal Pradesh tend to display brighter coloration, eggs, brownish, oval-shaped, and dorsally flattened. Larvae: the newly hatched larva has prominent black intersegmental markings on yellowish body and brown head. In the third instar, the prothoracic hood markings consist of two prominent, rectangular black marks which are replaced by semilunar, dark brown markings in the fourth and fifth instar. In Antheraea assamensis, for distinct color morphs viz. Green, yellow, blue and bright orange were earlier recorded, and except green form the other color morphs are very rare. The blue color larvae are bigger in size than the normal green form. The orange-colored larvae have a coating of white powdery substance on the body surface. Unlike the tubercles in green larvae, the tubercles of yellow larvae are white in color. The color of tubercles in green larvae changes during different instars: bluish in the second, purple in the third, brick red in the fourth and crimson red in the last instar. The outer color of the clasper transitions from fully black to fully green through partly black and partly green with increase of larval instars. Like the adults, the larvae of the wild strain in Arunachal Pradesh are more bright. Pupa and cocoon: the pupa is copper brown, while the single-shelled cocoon is golden brown, reelable, and slightly flossy, with a weak peduncle. Wild strain cocoons from Arunachal Pradesh exhibit brighter colors. Rearing of Antheraea assamensis: the Muga silkworm is multivoltine, having 5 to 6 overlapping generations annually. Egg laying and cocoon spinning are the only two stages of Muga silkworm rearing which are confined to indoor conditions. After the collection of seed cocoons, they are loosely packed in bamboo baskets containing dry straw and transported to the site of rearing during the night or early morning to avoid thermal shock. On reaching home, the cocoons are transferred to bamboo cages and stored in a thatched and mud-plastered house. Emergence of moths starts from dusk and continues till morning. 2 to 4 females are tied with thread around their forewing shoulder on a hook-shaped bundle of the culms of thatch grass. Imperata cylindrica called carica. The carica are hung on a frame. The male moths fly actively and mate with the stationary female. After overnight mating, the couples separate in the morning, and if they do not decouple naturally, they are made to do so by smoking, rotating a torch-like device made of straw, some distance away. After hatching of eggs, which need about 8 days during summer, the neonates along with the carica are suspended on the twigs of young trees. The tiny worms soon crawl up on the leaves and feed on them. The worms molt 4 times before they mature. When the larvae consume all the leaves of a plant, they are transferred to another plant using a triangular bamboo-made net called chelone. A special type of bow called darnu with round-shaped batalu guti prepared from clay is used against natural enemies like insectivorous birds and bats. The matured worms crawl down the tree during the night. A girdle of banana leaves usually is tied above the base of the host plant so that the downward larvae don't pass easily to the ground. The Muga picks them on a bundle of dry leafy twigs called the jali, the mountage. The mature worms are then taken inside the huts where they begin to spin cocoons within 3 to 5 days. After pupation and completion of cocoon spinning, the cocoons are removed from the jali. The best cocoons are sorted out and kept apart as seed cocoons for further propagation. The seed cocoons are kept in a rectangular, large cocoon cage made of bamboo or cane for moth emergence. Larval duration is around 1 month in summer and may last up to 2 months in winter. The pupal duration may last from 14 to 40 days and the adults from 7 to 14 days. Stifling means killing the pupa inside the cocoons so that the latter are not cut by the emerging moths. This is done first by spreading the cocoons on a bamboo mat and then exposing them to the sun. This partly kills the chrysalis. The mat is then kept above an oven where hot air and smoke completely kill the pupae. This process is repeated 2 to 3 times. Degumming means the removal of sericin protein covering the silk fibers that help to maintain the shape of the cocoons. This is done by boiling the cocoons in an alkaline solution of sodium carbonate for 10 to 30 minutes depending upon the amount of soda. 3 or 1 gram per liter present in the solution. Reeling. Muga cocoons are reeled on a primitive machine called beer in which 2 persons are needed, one to defloss the cocoon and draw filaments out of 8 to 10 cocoons and the other to twist the filaments and wound yarn on the spindle. Nowadays, improved machines that do the job faster are available.